Recombinant DNA, and the Biotechnology Revolution. From protein chains and DNA to higher levels of organization. Although Mendelian and molecular genetics represented important conceptual advances for evolutionary theory, the remarkable success of this conceptual framework also had a downside. The problem can be briefly stated as follows. Beyond biochemical pathways, most multicellular adaptations involve higher levels of organization that arise from complex interactions of macromolecules, not just single gene products. Additional principles of molecular, cell, and developmental biology are required to explain the relationship between the structure of genes and proteins and their useful functions. Unfortunately, the essential features of this information did not become available until later. One of the themes of this audiobook is the close relationship between evolutionary biology and an empirical understanding of the inner workings of the cell. Advances in both biology and medicine have played a major role in advances in evolutionary theory. Recombinant DNA technology is especially relevant since it provides insights into DNA, RNA, proteins, and various functions of the genetic apparatus. The power of recombinant DNA technology. The molecular structure of DNA, RNA, and proteins consists of polymers, chains of subunits, monomers, that are linked together in long chains by covalent bonds. Since similar sets of monomers, 4 different nucleotides for DNA and RNA, and 20 different amino acids for proteins, are used by virtually all species, DNA or RNA molecules transferred from a donor species to a host species will often perform similar functions in the host. This is also true for DNA and RNA sequences from viruses, which depend on host cells for their replication. A primer on the steps involved in protein synthesis can be found on Wikipedia. This also means that recombinant DNA sequences, DNA polymers derived from 2 different species, can be transferred into host cells, where they will be replicated, and where they can often be designed to synthesize mRNA and proteins in the host cell. The potential power of recombinant DNA technology was recognized by Herbert Boyer and colleagues at UCSF and Stanley Cohen and colleagues at Stanford University. These pioneers developed recombinant DNA technology in the early 1970s, which dramatically accelerated the pace of discovery in both biology and medicine. Briefly, recombinant DNA is prepared by inserting a desired sequence of DNA into a vector, usually either a plasmid, a small circular piece of DNA that can replicate in a harmless strain of E. coli bacteria, or the DNA of a virus, bacteriophage, that replicates in E. coli. Special enzymes, known as restriction enzymes, are used to cut the DNA sequences, and ligase enzymes are used to seal the DNA sequences together. The vectors are then grown in bacterial cell cultures, resulting in molecular clones of specific DNA sequences that can then be analyzed in a variety of ways. DNA sequencing, databases, and sequence alignments. Recombinant DNA technology has extraordinary power as a tool of discovery in evolutionary biology. As may be expected, the power of these laboratory techniques has been combined with several other technological advances, including DNA sequencing technology and sophisticated bioinformatics software for cataloging, searching, comparing, and analyzing massive amounts of sequence data.